Here is a very important scientific study that proves post-Avogadro diluted or molecular imprinted forms of drugs do not have inhibitory actions upon biological molecules, whereas their parent drugs in molecular forms can inhibit biological molecules. This study is very important in understanding the fundamental difference between molecular forms of drugs and molecular imprinted forms of drugs. According to MIT explanations, post-Avogadro potentized drugs contain molecular imprints of, which can act only upon pathogenic molecules having conformational affinity, and cannot interact with biological molecules, and hence, cannot produce any effect upon healthy individuals. Molecular imprints cannot mimic the properties of the crude molecular forms as some homeopathy theoreticians argue. Present research before us has experimentally validated this MIT view. This question whether potentized drugs interact with biological molecules in a way different from their parent drugs is very important in the scientific understanding of molecular processes involved in homeopathic potentization and therapeutics. There are many homeopaths believing that during potentization, the medicinal properties of drugs are some way or other transferred to the potentizing medium, and hence potentized medicines can interact with human organism in the same way as the original drugs do. On the contrary, only MIT proposes that potentization involves a process of molecular imprinting, in which the conformational properties of drug molecules are imprinted into the medium as three-dimensional intermolecular voids or molecular imprints, which can act as only as recognition sites or binding sites for original drug molecules or other molecules similar in conformation. As per this view, potentized medicines contain only molecular imprints of drug molecules, which are complementary in conformation to the drug molecules. When applied for therapeutic purpose, these molecular imprints bind only to the pathogenic molecules having conformational affinity, and not to the biological molecules. Molecular imprints thus remove the existing pathological molecular inhibitions, whereas crude molecules produce molecular inhibitions binding to the biological molecules. That means, molecular imprints do not act in a way similar to their original molecules when applied in a living organism. In order to prove this very important MIT concept, we have to experimentally prove that potentized medicines do not interact with biological molecules. Such an experimental proof is an essential requirement for proving MIT concepts. Here I am reproducing a previously published report regarding such an experiment already conducted by a team of eminent scientists in Germany a few years back. It is published in the Journal of Alternative and Complementary Medicine. May 2006, 12-4, 359-365. DOI 10.1089 ACM, 2006.12.359, http www.libet.online.com DOI ABS 10. 1089 ACM, 2006.12.359 Research team consisted of 1. Claudia M. Witt, MD Institute for Social Medicine, Epidemiology and Health Economics, Charité University Medical Center, Berlin, Germany. 2. Michael Bluth, MD Institute for Social Medicine, Epidemiology and Health Economics, Charité University Medical Center, Berlin, Germany. 3. Stefan Hinderlich, Ph.D. Institute for Biochemistry and Molecular Biology, Charité University Medical Center, Berlin, Germany. 4. Henning Albrecht, Ph.D. Karl and Veronica Carstens Foundation, Essen, Germany. 5. Rainer Ludtke, M.Sc. Karl and Veronica Carstens Foundation, Essen, Germany. 6. Thoralf E.R. Weishun Institute for Social Medicine, Epidemiology and Health Economics, Charité University Medical Center, Berlin, Germany. 7. Stefan N. Willich, M.D., MPH Institute for Social Medicine, Epidemiology and Health Economics, Charité University Medical Center, Berlin, German. The team conducted this experiment to verify whether potentized mercury 2-chloride Mercurius corrosivus affect the activity of diastase and alpha-amylase in a way similar to crude form of mercury 2-chloride. Their objective was to test for a stimulating or inhibiting effect of high potencies of the homeopathic remedy mercury 2-chloride Mercurius corrosivus on two sugar hydrolases, alpha-amylase from hog pancreas and diastase extract from winter barley. High potencies of mercury 2-chloride were produced using stepwise dilution plus shaking. Controls included potentized solvent aqua bidestilata, equimolar dilutions without shaking, and enzyme-free references. Tested were potencies with dilution factors 1 to 200 cc on diastase extract from winter barley, and 1 to 100 c on alpha-amylase from hog pancreas. Enzyme activity was colorimetrically determined by Lugol's iodine starch reaction. An inhibiting effect of mercury 2-chloride on enzyme activities was observed only in low potencies and dilutions which contained molecules of mercury 2-chloride. Statistically significant differences between potencies and controls were not found in randomized and blinded experiments. This experimental design provided independent reproducible results of cell-free in vitro assays. However, it did not indicate an effect of potentized mercury 2-chloride on hydrolases. The researchers' conclusion was that they could not demonstrate any inhibitory action of potentized mercury 2-chloride. Reported experiments and the results they obtained will help us in designing and conducting further in vitro experiments to prove the hypothesis put forward by MIT regarding potentization. HgCl2 is known in homeopathy as Merc-Cor. Crude mercury 2-chloride is a known inhibitor of glucose hydrolases such as diastase and alpha-amylase. Reported experiments show that similar to crude forms, lower dilutions of this compound also inhibits the hydrolyzing activity of those sugar hydrolase enzymes. Obviously, these lower dilutions contain molecules of mercury 2-chloride, and hence the inhibitory action on enzymes. Same time, these experiments clearly showed that higher potencies of mercury 2-chloride have no inhibitory action on those enzymes. That means, highly potentized mercury 2-chloride cannot mimic the original compound as expected by some theoreticians. This finding, though considered by the researchers as a setback to their expectations, has serious implications in proving the concepts of MIT regarding potentization. This experiment proves that through the potentization process, the properties of original drugs are not transferred to the potentizing medium in such a way so as to enable it to mimic the original drugs. We homeopaths know beyond any doubt that potentized mercury 2-chloride or Merc-Cor produces expected therapeutic effects when administered on the basis of principle of similia similibus curantur. That means, potentized mercury 2-chloride contains some active principles having specific biological properties. Since the present experiments have shown that potentized mercury 2-chloride cannot mimic the biochemical properties of original compound, a logical and scientific explanation regarding the real molecular mechanism involved in potentization as well as therapeutic action becomes very much necessary. Only possibility is molecular imprinting, as proposed by MIT. Now, we have to repeat these in vitro experiment to verify whether higher potencies of mercury 2-chloride can reactivate the enzymes already inhibited by lower potencies or crude forms of the same compound. Even though the reported experiment was not intended or designed to prove MIT concepts, it indirectly contributes in proving it.